16 viruses, designed by artificial intelligence, not predicted, not simulated in a spreadsheet somewhere, built, synthesized, sitting in a lab dish at Stanford right now, actually infecting bacteria, actually replicating, actually working. A few months ago we covered the model that made this possible, Evo, the genomic AI that could read and generate DNA the way a language model reads and generates text. Back then it was theory, a demonstration of what might be possible. Today it isn't theory anymore. Researchers from Stanford and the Arc Institute just published the results, and the headline is almost too clean to be true. They asked an AI to design an entire virus genome from scratch, synthesized nearly 300 of its designs, and 16 of them came alive. Before you picture something out of an outbreak movie, pump the brakes. These are bacteriophages, viruses that infect bacteria, not humans, not animals, not anything with a spine. They're the most common biological entity on Earth. They've been used medically for over a century, and the specific one at the center of this study is about as low stakes as a virus gets. So the fear-bait headline isn't the real story here. The real story is stranger, and honestly more impressive, than scientists made a scary virus. Here's the setup. The team, led by Stanford's Brian Hay, targeted a bacteriophage called FIX-174. If that name sounds oddly familiar, there's a reason. It's a legend in molecular biology. It was the very first genome ever fully sequenced, by Frederick Sanger back in the 1970s. It was also the first genome ever chemically synthesized from scratch, by Craig Venter's team in 2003. This tiny virus has been a proving ground for genetic technology for 50 years. And now it's the proving ground for generative AI. The tool doing the designing was Arc Institute's Evo Model Family, Evo 1, trained on 2.7 million prokaryotic and phage genomes, and its successor Evo 2, trained on 9.3 trillion nucleotides spanning the tree of life, built in collaboration with NVIDIA. Think of these as language models, except instead of predicting the next word in a sentence, they predict the next base pair in a genome. Feed them enough real DNA, and they start to internalize the grammar of life itself, which sequences fold into working proteins, which arrangements of genes actually cooperate to build something alive. So the researchers fine-tuned Evo specifically on over 14,000 genomes from the Microviridae family, the broader group FIX-174 belongs to, and then let it generate new genome designs, not fragments, not single genes. Full genomes, 5,386 nucleotides long, encoding 11 genes that all have to work together or the whole thing fails. Then came the part that actually matters, the part most AI biology headlines skip. They didn't just generate sequences and call it a day. They synthesized the DNA for real, inserted it into non-pathogenic lab strains of E. coli, and ran a high-throughput assay to see which ones could actually infect and kill their bacterial host. Out of roughly 285 designs put to the test, 16 showed genuine functional activity, a 5.6% hit rate. That number might sound low, but for a system designing an entire functioning organism from a blank page, with zero natural selection to clean up the failures, 5.6% viable is remarkable. Evolution had 4 billion years to get this right through trial and error. This model got there in a training run. And here's the detail that separates this from a party trick. These weren't just copies of natural phages with the serial numbers filed off. Each functional virus carried somewhere between 67 and 392 mutations compared to its closest known relative in nature. One design, the researchers labeled it Evo Phi 247, was still 93% identical to a natural genome, recognizable, but meaningfully novel. These are genomes that do not exist anywhere in the natural world, and never did, until a model imagined them and a lab brought them into being. Now here's the part that made actual virologists sit up. One of the designed phages, Evo Phi 36, managed to successfully borrow a structural protein from a completely different, distantly related phage called G4, and integrate it into a working genome. That's a move scientists have tried to pull off before with deliberate, rational genetic engineering, hand-designing the swap gene by gene, and it has historically been extremely difficult to get right, because swapping a protein from one viral architecture into another usually breaks something you didn't even know depended on it. The AI did it anyway. Not because it was told the rule for how proteins interact, because it had absorbed the pattern from millions of examples and generalized it to a case it had never explicitly seen. That's the actual insight buried under the 16 viruses headline. This isn't AI randomly guessing DNA sequences and getting lucky. The first author on the paper, Samuel King, put it simply, even at the scale of an entire genome, the model was able to reason about the different elements that need to work together to create something functional. That word, reason, is doing real work in that sentence. We're not talking about a system that memorized viruses and reshuffled them. We're talking about a system that appears to understand, at some level, the constraints that make a genome viable, well enough to design something new that respects those constraints. It's also worth being clear about how the team knew these 16 actually worked, because functional isn't a vibe, it's a measurable outcome. The primary test was a high-throughput growth inhibition assay. Basically, expose plates of the bacterial host to each candidate phage and watch whether it visibly suppresses bacterial growth the way a real working virus would. That's how the 16 were separated from the roughly 270 others that didn't make the cut. But the team went further than a pass-fail growth test. They used cryo-electron microscopy to actually image the structures these designed genomes produced, confirming that the AI wasn't just generating sequences that happened to look plausible on paper. It was generating instructions that folded into real, physically coherent viral particles. That's the difference between a model that's good at pattern-matching DNA syntax and a model that's implicitly modeling three-dimensional structure and function from one-dimensional sequence alone. Getting both of those right, at genome scale, without ever seeing the finished protein it's building, is the part actual structural biologists found hardest to believe. Zoom out and the application writing itself is obvious. Bacteriophages are one of our sharpest tools against antibiotic-resistant bacteria, the superbugs that don't respond to our best drugs anymore. Phage therapy already exists, but finding or engineering the right phage for a specific resistant strain is slow, manual, and often more art than science. If a model can generate viable novel phage genomes on demand, tuned to target a specific bacterial threat, that turns phage therapy from a slow custom tailoring process into something closer to a search and generate pipeline. That's the genuinely exciting version of this story, and it's not hype. It's the stated motivation behind the research itself. But we don't get to stop there, because the hype but true rule cuts both ways, and this story has a real counterweight that deserves equal airtime. The same capability that lets a model design a helpful, novel, harmless bacteria is in principle capability agnostic. It doesn't inherently know the difference between helpful phage and something far more dangerous. Biosecurity researchers at Johns Hopkins, including Thomas Inglesby and Moritz Hanke, flagged the actual vulnerability here, and it's not science fiction, it's an infrastructure gap. Right now, DNA synthesis screening works by matching requested sequences against databases of known dangerous pathogens. That system was built for a world where dangerous sequences look like other dangerous sequences we've already seen. A generative model doesn't have to do that. It can produce something with no evolutionary history, no fingerprint in any existing database, that still does what a bad actor wants it to do. The screening net was built to catch known shapes. These models can generate new ones. To be fair to the researchers here, and this matters, they clearly took that risk seriously. They worked exclusively with non-pathogenic E. coli host strains in properly contained lab conditions. And critically, the training data itself deliberately excluded human viral sequences, specifically to make it harder for a model like this to be pointed at anything that could infect a person. That's a real safeguard, not a footnote. But safeguards at the research level don't automatically become safeguards at the policy level. Right now, in most places, you can order custom synthesized DNA online for a fraction of a cent per base pair, with no legal requirement that the synthesis company verify who's ordering it, or run function-based screening on what it actually does. There's a pending bill in the US, the Biosecurity Modernization and Innovation Act, aimed at closing that gap. But critics point out, it still leans heavily on the same sequence matching approach that this exact research just demonstrated a workaround for. So where does that leave us? This is, without exaggeration, one of the clearest demonstrations yet that generative AI has crossed from predicting biology to authoring it. 16 functional, novel viral genomes, born entirely inside a model's weights, validated in a real lab, with at least one genuinely creative engineering trick, the G4 protein swap, that human designers have struggled to pull off deliberately. That's the frontier moving in real time. And it comes bundled with the exact tension that defines every powerful new tool in this field. The same reasoning that makes a model brilliant at designing something useful is the same reasoning that makes oversight infrastructure race to keep up. This research was published as a preprint in September, meaning it hasn't finished formal peer review yet. Worth remembering before you treat every number here as final. But preprint or not, the phage in the dish is real, and it's working. If you've been following this channel, you already know the pattern by now. The gap between AI can generate it, and AI-generated things exist in the physical world, functioning, keeps closing faster than most people expect. Biology just became the newest place that happened. Subscribe if you want the next one the moment it lands, because at the rate this field is moving, there will be a next one soon.